Jennell. I'm a second year PhD student in the Biomedical Sciences program here at UCR. My lab primarily studies innate immune responses to infectious diseases. This is a day in my life. So how I like to start my day is I usually like to get settled in, maybe have a cup of coffee, and get my emails done. So as a graduate student, you have a lot of communications that you have to do and interacting with other scientists as well as just other people in general. That's usually how I like to start my day. I also like to start my day by getting any other computer work done, and that includes analyzing data, making graphs, etc. In my lab, we study innate immune responses to parasitic infections. And so one thing that I like to do, or that we do in our lab, is we make these cytosins here. So what these are cells isolated from the lungs of an infected mouse. We work primarily with hookworm. And here what we like to see are pretty much a smear of the cells together with the worm. And this is a really good way to look at the interactions between the parasites and the worms that we collect from our lungs. To view these slides, you just like to take a quick look under the microscope here, focus, and make our way around each of the slides.
Can you take a look?
Yeah, of course. So within the slide, I can see cells, also known as eosinophils, and these are the main effectors against parasitic infections. They are also what causes asthma and allergic inflammation, so you can blame these little guys for that.
So where are we going next?
So next we're going to the cell culture room. So your body is made up of millions and millions of little cells. You have skin cells, eye cells, hair cells, all sorts of cells. And in cell culture, what we can do is we can isolate these cells either from a human or a mouse, and then we can continue to culture them or grow them using artificial means within the lab.
Cool, let's see.
Welcome in. So this right here is what we call a biosafety cabinet. So this is what we use to make sure our cells stay sterile. It has a filtration system that pushes air out and filters it before it comes back in, and this is what prevents any sort of bacteria, viruses, fungi, or any sort of nasty bug from getting in here and getting in contact with your very precious cells. Our cells stay super sterile. We're going to spray our hands down and the hood down with 70% ethanol. So this is super great because it's similar to another alcohol that's used in hand sanitizer, but generally these alcohols work to kill any sort of bacteria or anything that may be on your hands before you start doing your cell culture. What we're going to do is our cells are going to be attached to the bottom part of this flask. This flask has a special charge on it which allows our cells to actually sit down on this flask so that we can add and change media without disturbing our cells. We're going to use this. This is also known as an autopipette or a serological pipette. And this is what we can use to suck up all the stuff that we need to feed our cells. So right here, we have cell media. So this is essentially Kool-Aid for cells. We have a lot of sugars in here, salts, and other essential nutrients that we're going to use and that we need in order for our cells to be happy and healthy and continue to grow. This even has a special filter here on the tip, and that's going to help our cells get oxygen and the carbon dioxide they need to grow healthy. Welcome to our microscope room. Here we have this super huge microscope that's known as the Keons, and what this does is allow us to image all of our worms and all of our cells, especially when we do cult cultures, to look at the interaction between parasitic worms and cells. Open it up. Inside we have a huge lens here, and we also have a lens system below here with lighting. This microscope is able to see emissions from different lasers, so you can also complete immunofluorescence staining, which is when we look at specific markers on cells to determine what cells they are and what they do. So as you can see here, we have all of our nice squiggly, squirmy parasites along with some cells called macrophages. Macrophages are another type of immune cell that are the main effectors against fighting against parasitic infections. They're also really important for fighting bacterial infections as well. So here we are able to really clearly see the interactions between these really cool cells and parasitic worms. This is our life sciences incubator. Here we have a whole bunch of startup biotech companies that are doing scientific research to help the newest advancements in pharmaceuticals, medication, and other subjects as well. UCR provides many really cool instrumentations for these startups to use in order to help advance their scientific research. Here we have anything from PCR machines. This helps us look at different genes that are upregulated or downregulated, and this helps to show what is happening in our bodies and how we can use this information to advance medical research. This right here is a flow cytometer. Flow cytometers are used in order to look at different proportions of cells when in a certain medium. Is also used to look at different upregulation of certain markers and genes within these cells. Every day I like to come down here to run different experiments. This is open to both the startup biotech companies in here as well as UCR laboratories. So grad students are welcome to use this as well as the companies that rent this place. So within here we have many different instruments. Right here, this is a water filter. This gives us super crispy, clean water that we need to use for research. We also have titrators here. This is to help us with determining the pH and titrating different chemical solutions to match the pH that we want. These over here are chemical scales that are super precise. And then we also have thermal cyclers. These are really important again for running our PCR, our Bloom reaction. This is really also important for genotyping, which is ensuring that certain mice strains are the ones that we want. This can also be for human research as well. So this is a really important machine for genetic analysis. I like to come in here and chat with other grad students or individuals who work in the companies that utilize this space. My day also consists of analyzing data and making graphs that will later be used either for scientific presentations that I may have, or we may be able to put it in a scientific publication of our own. So a big part of our day is putting data into the computer and then analyzing it and getting graphs that we can later use to display our science to other people. It also consists of analyzing images that we get from some of our experiments. This is a slice that we took from an infected mouse lung. So again, our lab likes to study the interactions between parasites and immune cells and how that these parasites contribute to tissue damage and how our immune cells can help in terms of making sure that we heal from that tissue damage. Part of being a grad student is writing grants. Grants are really important so that our projects that we like get funded. So as part of writing this grant, we like to look at other preliminary data that we may have that may support our hypothesis, and then we also like to speak about the background and significance of our project and why it's important that this research is undergone. After finishing a little bit more of my lab work, a really important part of being a grad student is making sure that you're up to date on the newest scientific advancements and discoveries. So how I like to spend my day usually is reading scientific journals that are very recent, and so I can learn more about what's going on in science today. So as a PhD student, we still are required to take courses. However, our courses are much more defined and much more specific to what we're going to be studying and doing for the rest of our lives. One course that we have to take here at UCR is a review writing course. For this review writing course, we look at other scientific journals similarly to what I do every day, and we like to do an overall summary of what these journals say and the advancements to a specific field in science. We also take other courses, including basics to human physiology and basic science courses such as biochemistry and immunology as well. Hi, I'm Jennell. I'm a second-year PhD student here at UCR. I study innate immune responses to infectious disease, and this is my story. So I've always had a really keen interest in the sciences. When I was younger, I always liked to go outside and look at leaves, look at rocks. It was always something I really enjoyed doing. I knew at a young age that science was definitely the route for me, and I really couldn't imagine myself doing anything else in life. I come from an immigrant family. Both of my parents are immigrants from Belize. I'm a first-generation American. Neither of my parents attended college, so I was the first person in my family to attend college. Starting out, like I said, I always knew I wanted to go into science. However, I wasn't really sure how to do that and how to get there. When I was in middle school, high school, I just made sure to take all the science courses that were available to me. I was also part of leadership of different scientific clubs, one of those being HOSA, or Health Occupation Students of America, which got me a lot of networking opportunities with people in the sciences, and really gave me the first step that I needed to really figure out what I needed to do to achieve my goals. I soon went and studied my undergraduate degree at Arizona State University. There I studied microbiology. I mean, it was really tough. I didn't really have a lot of background in what I should be doing, what was going to happen, and it was really just taking it step by step. I'm so grateful for the faculty members who truly believed in me, who really helped me when I wasn't really sure what else to do. I had my first research experience actually in a honeybee laboratory, and where we studied olfactory systems in honeybees, and we actually did honeybee training. So my first research experience was not in immunology whatsoever, but it was a really good experience, and it really taught me a lot of how to do science, how to get data, how to analyze data, everything. So it was really helpful. I soon graduated from undergrad, but I was still unsure what I wanted to do. And I think a lot of the times, you know, when you're interested in science, people say, go and be a doctor, go and be a physician. And I wasn't quite sure what I wanted to do, but it was something that interested me. So moving forward, I decided to actually get a job as a medical assistant at a local primary care clinic, and soon I realized that that wasn't really for me. I really enjoyed the science and the research more than the front-facing interaction. And so moving forward, I decided to actually get my master's degree. So I completed a one-year master's degree in Tampa in medical sciences, and at that point I knew that a career in science is what I wanted to do. So actually, after I finished my master's degree, I moved to San Diego and I got a job in the biotechnology industry. And this was probably the biggest part of really what made me want to come and pursue my PhD. I got to study a lot of amazing things in the biotech industry. I got to study anything from breast cancer. I also worked on psoriasis medications. I got to work in stem cells. And so it was a really great opportunity for me to get a lot of background information for a lot of different subjects within science. And at that point, I really knew that biotech was what I wanted to remain in. However, given my degree, I was somewhat limited to the positions that I could obtain. So it was actually my mentor and my manager in biotech that really encouraged me to maybe pursue a PhD, so that I could have more independence in the lab when I did decide to return to biotech. So that brings me to today. I decided to apply for my PhD, and I was lucky enough that my mentors within biotech really helped me in terms of getting my application together, what PhD programs are looking for. And I really recommend to really use all your resources that you have available to you. Although my parents didn't come from a background where they were able to help me, I was able to find others around me, either at my school or at any of the jobs that I worked, that really took the time to really get me to a place where I felt comfortable and that I understood what was going on and what I needed to be doing. And even to this day, I'm still not sure everything that I need to be doing, but it's something that you take a step at a time. And if you have the motivation, if you have the dedication, you have the passion, people will see that and people will want to help you. And now I'm a step closer to getting the career of my dreams. I love what I do every single day. I get to do science, which, you know, it's crazy that it's been able to be a job for me when I just, before I saw it as something just fun and something that I like to do and I was passionate about. And so my advice to any other first generation students out there is to really network, really reach out to people. People really do want to help you. And when you reach out and you say, hey, I'm passionate about this, I'm just really not sure what to do, people are usually receptive to that. And so go out there. Social media is huge. If you see, you know, people out there in careers that you like, or, you know, have friends or family members, you know, distant family members that may know someone who knows someone, you know, any way that you can network is really great in terms of giving you that background information researching online, going on forums, all of those things really helped me in terms of getting myself established into a place where I can make all of this happen. And even then, that's not to say there wasn't hiccups along the way. It's very common to make mistakes, especially when you're the first one in your family to do something like this. It's very common to make mistakes. So don't be too hard on yourself, and you're doing a really crazy big thing. You're doing a great step. So keep on doing what you're doing and you will find success.